we mainly focused on three most common monogenic cerebral small vessel disease, which is a disease that typically presents as which typically in patients it presents with stroke and dementia. And in our study, it's basically based on a previous paper that was published by Julie Rutten in 2018 that showed that these monogenic variants are causing small vessel disease were more common than expected in the general population. And then we wanted to assess whether this is also the case in the UK population. And in our study, we mainly focused on using the UK Biobank data, which is a data set of almost 500,000 individuals who are apparently asymptomatic. So most of these patients wouldn't have any major symptoms when they were recruited. And we look at these monogenic variants for cSVD in this group of individuals and see how common they are in the general population, and also whether they are associated with any disease like stroke and dementia. And we also, I guess the second aim is also to try to assess what is causing or influencing the variability in the phenotype of the individuals carrying these mutations, and so using the exome sequencing data in the UK Biobank, we try to correlate the presence of these variants with electronic health records and brain MRI scans measures, and then we found that in our study that these variants are actually indeed, and confirmed that previous finding that the variants are actually more common than expected. And these variants are associated with disease like stroke and dementia. And we also found that cardiovascular risk factors and the location or position of these mutations also influence the severity of carriers of these monogenic mutations. Traditionally speaking, it was thought that little can be done for people carrying these pathogenic variants that are so associated, familiar with the conditions or monogenic conditions. But I think a major finding in our study is that it actually suggests that maybe we can control cardiovascular risk factors, or clinicians can pay more attention to basic clinical details such as controlling blood pressure, or maintaining a healthy lifestyle, and exercising would actually reduce your risk of stroke, or mitigate your risk of stroke and dementia, and kind of offset your inherent genetic risk for these conditions. And that's one thing. And I guess it's still it's very tempting to say that controlling cardiovascular risk factors would lead to treatment response or reduce stroke burden. But I guess this study provides a foundation for further studies to understand what is causing this reduced or increased in stroke risk, and how these individual cardiovascular risk factors are interacting with pathogenic mutations to cause stroke and other neurological conditions. There are actually, I think, a few groups are working on, try to moving move forward from this study. And I guess first thing is because the UK Biobank is mainly focused on European population and UK population, we'd also want to know whether our findings would translate or would be able to replicate these findings in Asian populations, especially because these monogenic variants causing SVD have been found to be more common in Asia, and for example, in Japan, it's been found the majority of the HTRA1 associated SVD is found in Japan, and we also want to know whether these Asian populations will also have more common these variants are more common in these Asian populations. And secondly, it's also to try to elucidate what's going on behind the scene, and what's the molecular mechanisms associated with these monogenic variants, and interaction between monogenic variants and the cardiovascular risk factors and causing stroke. And yeah, so a lot of studies yet to be done and can be done to try to, find ways to benefit patients' population.